Yes, ma'am.
Good evening, everyone. Myself, Dr. Aanchal, and today we're going to start with the new topic in this series, that is WBCs, right? So first of all, let's see some core feature of Oracle. So in Oracle, we provide recorded classes, live classes, practical sessions, exam module, and also we provide hard copy book sets. Recorded classes are actually for the elevated learning of the student so that the student can have the access of all the subjects anytime, anywhere through the recorded sessions. Then also we provide live classes so that the depth coverage of the topic can be studied properly. Then exam modules are there so that the student help the can have the help from those exam module, and they can score well in the examination. And also we one-to-one doubt clearing sessions are there so that the student can ask doubts anytime, anywhere in Oracle. Then practical sessions are there, and as you know, our expert educators are there to guide you through the Viva, as well, ensuring that the student gain the skills that are needed for the examination. Then question banks are there. In question bank, we provide questions from beginner level to expert level, and which contain standard multiple choice questions. Then hard copy sets are there. So in hard copy sets, basically this is designed to cover the entire syllabus, and all the important topics will be there, including past 10-year asked questions. We provide subscription plans starting from basic to premium plan. So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. In premium plan, we provide all the services like pre-recorded classes, live class, live doubt clarification, one-to-one personalized guidance, notes, quick revision examination module. And this plan cost 59,999. And also for instant 20% discount, you can use my code, that is Dr. Aanchal 20. So please subscribe to Oracle as soon as possible. So in today's class, we're going to learn about the leukocytes, and then we're going to know about the development and function at different leukocytes. Okay. So if we talk about the leukocytes, so what are leukocytes? Leukocytes are WBCs, right? Leukocytes are WBCs, which are colorless and nucleated formed elements, right? So WBCs are colorless and nucleated formed elements of blood, right? So compared to RBC, the WBC have larger sizes, but they are in lesser number. Okay. So talking about the count, so the count range between 7,000 to 11,000. Okay. Then talking about the lifespan. So lifespan is also shorter as compared to RBC, because RBC has 120-day lifespan, but the lifespan of WBC is of only few weeks, right? Now, coming to the classification, okay? So we know we classify the WBC on the basis of presence or absence of granules. Like in the WBC in which granules are present, they are known as granulocytes. The WBC in which granules are absent, so they are known as agranulocytes. In granulocytes, we have basophils, eosinophils, neutrophils, then monocytes and lymphocytes. Okay. Now, coming to the various stages of leukophysis means how the development of WBC take place. Okay. So, various stages of leukophysis we're going to study with the help of a chart. Okay, I'm going to draw a table for that, and I hope you will have the better understanding with that table. Okay, so we know that the first cell is the mother cell from which all the cells derive. Okay, so first cell we have primitive WBC, or we can call that it is a stem cell. Then next we have myeloblast. Then next we have myelocyte. Okay, but this is myelocyte A. Then we have myelocyte B. And then finally we have myelocyte C. Okay. And after myelocyte C, finally we get the mature WBC. Okay. So these are the various stages from which we get the mature WBC derived from the stem cell or derived from the primitive WBC. Okay. Now, if we talk about the difference, okay, and what are the development stages are there? What about the cell size, nucleus, cytoplasm? What about the division? So, how actually the changes take place through the various stages that we have discussed. So, if we talk about the cell size, okay, so these are the stages. Then, if we talk about the cell size, right? So cell size of WBC decreases as the development of WBC takes place. Okay. So cell size like the primitive WBC has cell size of 18 to 23 mu m. Then myeloblast will be 16 to 20 mu m. Then myelocyte A 14 to 18. Then myelocyte B 12 to 16. Then myelocyte C 10 to 14. And finally, the mature will have less than 10, that is approximately 9 mu. Okay. Now, talking about the next characteristics means what about the nucleus? Okay. So, if we talk about the nucleus, so obviously, as the size of the WBC will decreases in further stages, so obviously it will lead to decrease in the size of nucleus too. Okay, so that we're going to see how. So here we have nucleus, then we have cytoplasm, and in others we're going to talk about the cell division. Fine. Okay. So first we're going to talk about the nucleus. Okay. So in the primitive WBC, the size of the nucleus will be very large. Okay. And many nuclei are there. Okay. Then in myelocyte also, the size of nucleus is large. Again, nuclei are there, but the size will be small as compared to the primitive WBC. Then, starting from the myelocyte A, the size will starting decreasing. Okay, in myelocyte B, the size become round and nucleus size further decreases. Then in myelocyte C also, the nucleus size decreases further, right? And finally, in mature WBC, the size will become more less. Then talking about the cytoplasm, so as the size of the nucleus will decreases, so size of the cytoplasm will start increasing. Okay, so here in primitive we have basophilic thin rim of cytoplasm, right? And the amount will increase, and it will continue on increasing. But in myelocyte B, there will be appearance of granules suddenly. And in myelocyte B, it will start, in myelocyte C, it will start showing the movement that is known as amoeboid movement. Right? So this is all about the size of the cytoplasm. Now next we have mitosis. So in primitive WBC, as cell division is present, so mitosis will be there. Then mitosis is present in myeloblast also. Then in myelocyte A also. Then multiplication will be maximum in myelocyte B. And finally, in myelocyte C, the mitosis will stop. Okay, so this is how actually the various stages takes place in the leukopoiesis. Okay, so I hope this is very well clear to all the students, right? Now, next we have describe leukocytes, morphological features with the help of diagram, elaborate the steps involved in the phagocytic function of neutrophil. So now we're going to know about the different leukocytes with their morphological features. Please close the door. Close. Door close. Okay. So first we're going to know about the neutrophil. Okay. So neutrophil is also known as polymorph because it have many many lobes are there. It is basically multi-lobe and the lobes contains between 2 to 7 lobes. Okay, so basically the number of lobes is representing the life, that how long the neutrophil has become old. Okay, now basically the number of lobes you can say that in the nucleus depend upon the age of the cell. Okay, so in younger cell the nucleus is not lobed, but in the older cell the lobes can be range from 2 to 7. Okay, then next we have eosinophils. So eosinophils has a pores larger granules in the cytoplasm. Granules are very large with stain pink or red. Okay. Nucleus is bilobed like this. Okay. And the diameter is range between 10 to 14 mm. Then next we have the basophil. So basophil have also pores granules. The granules is stained purple blue with methyl blue, and the nucleus is again bilobed and it is basically of bean shaped like this. Okay, again this is bilobed. Then next we have monocytes. Okay, so the monocytes are the largest nucleoside with a diameter of about 14 to 18 mu, and the cytoplasm is very clear because no granules are present, and the nucleus is horseshoe shaped. This is the identification features of the monocyte. Okay, then last we have the lymphocyte. Okay, so in lymphocytes, the lymphocytes contain the very large nucleus covering almost 3/4 part of the this cytoplasm. Okay, so it has clear cytoplasm and nucleus occupying whole of cytoplasm. So in horseshoe shaped, it will be like this, like this. Okay, so these are the identification features of all the various WBCs. Okay, now next we're going to understand the process or steps involved in phagocytosis. Okay, so when we talk about phagocytosis, phagocytosis means the process by which particles larger than the macromolecules are engulfed into the cell. Okay, so these are various steps. First step, second step, third step, fourth step, fifth, and finally sixth. Okay, so one by one we're going to understand all these steps. Okay, so as we have discussed, phagocytosis is the process by which particles larger than the micromolecules are engulfed into the cell, and this is also known as the cell eating process. Okay, so basically larger bacteria, larger antigen, and any larger foreign bodies are taken inside the cell by this process that we know that is known as phagocytosis. Okay. Only few cells in the body, like neutrophils, monocytes, and the tissue macrophages, show this process of phagocytosis. So among these, the macrophages are the largest phagocytic cells. Okay. Now we're going to understand the mechanism. The mechanism is very important. So as we can see in the slide also, in first picture, the cell is there and the pathogen is there, which need to get engulfed by the cell. Okay. So what happened when bacteria or any foreign particle, foreign body entered the cell? First, phagocytic cell send cytoplasmic extension that is known as pseudopodia. So that extension will be like this. Okay, so that will be around that particular pathogen. Okay, now these particles engulf and are converted into like a endosome that is known as vitriol. Okay, so they will form the endosome like this. Okay, so first they will detect this, then they will project out pseudopodia, they will engulf or they make an envelope around it, then the process of digestion will get started. Okay, so the phagosomes are there that travel into the interior of the cell, and primary lysosomes are there that fuse with phagosomes, and second they form the secondary lysosomes. Okay, so first there will be formation of primary lysosome, then secondary. As we can see, they have mentioned a complex that is known as major histocompatibility complex. So with the help of this, now what will happen? The pathogen break down into protein and other molecules. Okay. Now what will happen? How this breakdown has occurred? This breakdown has occurred with the help of some enzymes that are known as hydrolytic enzymes. Okay. That result in digestion and degradation of the phagosomal content. And finally, with this process, it will lead to elimination of the bacteria or pathogen which has entered into this body illegally. Okay, so this is all about the entire process of phagocytosis. Again, I'm repeating how it's working. So phagocytosis means the larger particles that they can be eliminated out from the body. So first, the cell will detect that pathogen, then they will project out pseudopodia. They will form with the like envelope they will form, then digestion will perform with the help of hydrolytic enzyme, then that will break down into protein and other molecule, and finally elimination will occur. Okay. Now, next we have the macrophages. Okay. The function of macrophages is very important. You need to understand. So, basically, what are macrophages? Macrophages are also a type of red blood cells of the innate immune system that engulf and digest pathogens such as cancer cells, microbes, cellular debris, foreign substances. But they do not have that protein that are specific to healthy body cells on their surface. Okay, so I'm writing that macrophages are also a type of WBC of the innate immune system. Okay, so they are the WBC cells that plays an important role in the immune system. Additional role within the specific organ system, they engulf and digest macromolecules, clear out debris and dead cells, stimulates other cells involved in the immune function. Okay. Now, another thing that the majority of the macrophages are stationed at the strategic points where microbial invasion or accumulation of the foreign particles is likely to occur. Okay. Now, coming to the function part. So, macromolecules are professional phagocytes. Okay. So, the macrophages are phagocytes. So, if they are phagocytes, so means they are involved in the process of phagocytosis also. Okay. Also, they play a very important role in chronic inflammation. Okay, at the early stages of inflammation are dominated by neutrophil, which are ingested by macrophages if they come of age of a certain period. Okay. Now, the neutrophil are at first attracted to a site where they perform their function and die before they or their neutrophil extracellular traps are phagocytes by the macrophage. Okay. So overall, if we look into the picture, so macrophages are those specialized cells which are very, very important for performing the function of phagocytosis. The next we have the difference between T and B cells. Okay. Now, another thing that the difference between T and B cells is very important, and usually the question over the T and B cells come. Okay. So we're going to understand the difference between T and B cells again by drawing a chart. So T cells basically originate in bone marrow and mature in thymus. That's why they have named this as T cells. Okay. So originate in bone marrow and mature in thymus. Whereas the B cells originate from bone marrow but mature in again in bone marrow. Okay. Now, the T cells, mature cells, they are the mature cells that occurs inside the lymph nodes. Okay. So, they are mature cells that occurs inside bone. B cells are mature cells occur outside the lymph node. Okay. Then these T cells bear TCR receptors. And the B cells are responsible for bearing BCR receptors. Okay. T cells have long lifespan as compared to B cells. Okay. So they have long lifespan, but the lifespan is shorter as compared to T cells. Okay. So, the T cells are responsible for the secretion of lymphokines, whereas the B cells are responsible for the secretion of antibodies. Okay. So, this is a very important difference between T cell and B cells, that they secretes lymphokines and the B cell secretes antibodies. Okay. Now, talking about the concentration, that in majority which cells are present and in minority which cells are present. So T cells are 80% of total lymphocytes, whereas the B cells only 20% of total lymphocytes. Okay. A very important and another difference, that T cells are always act against tumor cells or transplant, whereas the B cells do not act against tumor cells or transplant. Okay. So this is a very important difference. All the students should note it, that the T cells act against tumor cells or transplant. The B cells do not. Okay, so these are the major difference that should all the students learn between T and B cells. Okay, so once again, I'm going to repeat all the difference that we have learned. So first difference we have learned that T cells originate in bone marrow, whereas mature in thymus. B cells originate in bone marrow and also maturation will occur in thymus. In bone marrow. Then in T cells, the mature cell occurs inside the bone marrow, and in B cells, mature cells occur outside the lymph node. Then T cells bear TCR receptors, B cells bear BCR receptors. T cells have longer lifespan, B cells have shorter lifespan. T cell secretes lymphokine and B cells responsible for the secretion of antibodies. T cell is represent 80% of total lymphocytes, whereas the B cells are responsible for only presenting 20% of total lymphocytes, and T cells act against tumor cells or transplant, whereas the B cells do not. Okay, so these are the major difference between T cells and B cells. Okay, fine. Now, next we're going to learn about the functions of WBCs. So we know WBCs are leukocytes and they are colorless. So we're going to learn these four functions which are very important. So they protect the body against the allergic parasitic infection. Another thing that they secretes and transport are very important substances like heparin, histamine, and the serotonin. Another thing that the WBCs are phagocytic in nature, so therefore they destroy the various pathogen, bacteria, viruses which illegally enters inside the body. So the elimination will occur by the process that is known as phagocytosis. The WBCs are also known as scavengers of blood. Why they are known as scavengers of blood? Because they engulf, damage, or dead and minute bits of blood cells. Right? So this is all about the major function that is performed by the various WBCs that are present inside the body. Okay. Then next we have the another important process that we're going to understand, that is chemotaxis. Okay. So what is chemotaxis? Basically, bacteria products interact with the plasma protein to produce an agent, and that attract neutrophil to the site of injury or inflammation, that is known as chemotaxis. Okay. So, as a result, chemotaxis, neutrophil adhere to each other, forming the clump and become immobile. And the chemotactic agents are, various chemotactic agents are there, like so chemotactic agents includes components of complement system, that is from C5A, then some leukotrienes are there, and also colony stimulating factors are there. Then toxins are there that produced by macroorganism. Last, the kinins produced by damaged tissues. Okay. So these all are the chemotactic agents that are responsible for this process of chemotaxis. Okay. Now, next we have the monocytes. We're going to know about the morphology and its function. Okay, so when we talk about the monocyte, so already we have learned that monocyte is also a type of WBC, which comes under a granulocyte. So now we're going to know about the little bit about this morphology and also about its function. And this is the very important topic because it has previously asked in some university exams also. Okay, so if we talk about the size of monocyte, so size is around 10 to 18 mu with irregular cell outline. Okay. Then talking about the nucleus. So the nucleus is pale staining, kidney shape, then coming to cytoplasm. So cytoplasm is usually pale blue and appears clear. Okay. Then talking about the granules. Sometimes contain fine purple dust like granules that are known as azure granules. Okay, which may be few or which may be numerous. Okay, fine. Now we're going to talk about its function. So First, we have function that it participates actively in the process of phagocytosis. Okay, so monocytes follow the neutrophil in the area of infection or inflammation, and it is responsible for constituting the second line defense. Okay, so it constitutes second line defense. Then third, monocytes enter the circulation from the bone marrow, but after 72 hours, they entered the tissue to become tissue macrophages. Okay, so it acts as the tissue macrophages also. Okay, then the monocyte may also kill tumor cells after sensitization by lymphocytes. Ayush, get band kar. Okay, so monocytes is responsible for killing tumor cells also. Okay, then it is also responsible for synthesizing the complement and other biologically important substances like prostaglandin E and clot promoting factors. Okay, so it is responsible for the synthesis of complement like complement important substances like prostaglandin E and clot promoting factor. Okay, fine. Now we're going to talk about the monocytosis and monocytopenia. So first we have monocytosis, then we have monocytopenia. Okay, so monocytosis means there will be increase in monocytes. So increase in monocytes occurs in the cases like tuberculosis, syphilis, in case of viral or fungal infection, then due to chronic disease. Okay, then next we have the monocytopenia means decrease in monocytes. And decrease in monocytes occurs due to hypoplastic bone marrow. Then it could be due to other reasons also whenever there is a depression in bone marrow. Okay, so various other reasons are also there. Okay, so this is all about the WBCs. Again, we're going to revise it once again. So we have learned about the leukocytes. In leukocytes, we have learned that the WBCs are those colorless and nucleated formed elements which are very important for the defense mechanism inside the body. Okay, so we have learned WBCs are classified as granulocytes and agranulocytes. In granulocytes, we have basophils, eosinophils, neutrophils. In agranulocytes, we have monocytes and lymphocytes, right? Then we have learned about the various stages, like starting from primitive WBC, myeloblast, myelocyte A, B, C, and finally mature WBC. So as these stages proceed, the cell size will decrease, nucleus will also decrease, cytoplasm will increase, and mitosis will stop in myelocyte C. And also in cytoplasm will have the granulous appearance in myelocyte B, and it will start showing the amoeboid movement in myelocyte C. Okay. Next, we have the different leukocyte. We have understand like neutrophils, eosinophils, basophils, and monocytes and lymphocytes. Like neutrophil have many lobes, eosinophils have base lobe, bilobed, then basophil have bilobed, bean shape or kidney shape. Monocytes are like horseshoe shaped, and lymphocytes are having very large nucleus. Okay. Then next we have the phagocytosis. So phagocytosis means cell eating procedure. Yeah, means if the any foreign particle, large macromolecule entered inside the body through any illegal way, so the body will try to eliminate with the help of this process known as phagocytosis. Then we have learned about the macrophages function. We have learned that it is a type of WBC of the innate immunity system, and this is very, very important for the process of phagocytosis. Then next we have learned the difference between various T cells and B cells. And finally, we have learned the various function of WBC, which is a very important question asked in the previous year question also. Then the remote access we have learned, and in the last, we have learned about monocytes. Okay, so this is all for today's class. I hope you find this class helpful. For any kind of doubt and query, you can connect to us with the various social media platform like YouTube, Instagram, Facebook, and Telegram. So thank you so much for watching my class.